Yeah, today I will present how UAV-based phenotyping can be used to estimate sorghum lodging and assess its applicability in current uh, plant sorghum breeding trials. Sorghum production in Australia is highly variable uh, from year to year due to seasonal rainfall. Sorghum is a rain-fed crop grown on clay soils with high water holding capacity. Farmers usually plant only when enough water has accumulated in the soil profiles. If rainfall is limited later in the season, the crop runs out of water during grain filling stage. This later season uh, stress is called terminal water stress. It is a major driver of lodging. Lodging is a significant challenge in sorghum as it is in other cereal crops. However, unlike many other cereal crops, sorghum can remain alive after grain filling stage if water is available. This makes the expression of lodging more complex. In Australia, sorghum growing environments, lodging often occurs when plant express terminal water stress. Terminal water stress reduces photosynthesis during grain filling stage. As a result, carbon is remobilized from the stem to the panicle to fill the developing grain. This weakens the stem, contributes to the plant death, and increase the risk of lodging. Therefore, lodging is a major cause of both direct and indirect yield loss for growers. The direct yield losses are very obvious due to the inability of the harvester to pick up the grain on the ground. However, there is another important concern, which is indirect yield loss. Farmers may avoid high, high yielding hybrids if they have higher chance of lodging. Over many years, this can reduce average yield because farmers may choose safer hybrids with low lodging risk, even if those hybrids have low average yield potential. Genetic improvement requires consistent trait measurement. Plant breeders need to able to measure lodging at scale. Plant breeders currently rely on visual scoring approach to measure lodging, which is typically time consuming, expensive, and may be inaccurate. A practical alternative is measuring lodging using UAV imagery, which can be high throughput, lower cost, and may increase accuracy of measuring lodging. To do this, we collected phenotypic and UAV imagery data from 3 different sites. Each site contained 2 trial series. Uh, for this study, we had 3 types of the data available. For the purpose of my PhD research, we used manual counts where we counted the total number of plants and number of lodged plants in each plot and converted the result into a scale from 0 to 100. Manual counting is very labor intensive and is not used in practice. However, it is potentially more accurate. Breeders typically use visual scoring approach in current breeding programs where the breeder visually rates for each plot for the degree of lodging on a scale from 1 to 9. Uh, the 1 represents 0 to 11 percentage of lodging. The 9 indicates 89 percentage to 100 percentage of lodging. The manual counts and visual scores are positively correlated. The third data type was UAV imagery. I am developing an approach to measure lodging using UAV imagery. To do this, I used 2 sites with manual counts and UAV imagery data, then developed a framework. That developed framework was validated at a site where we have UAV imagery and visual scores to see the broad applicability of this framework in current plant breeding programs. To develop the approach using UAV imagery, the drone with a multispectral camera flown over the trail following a planned flight path. During each flight, the drone captured thousands of overlapping images from different angles. Those images were imported into photogrammetric software where they were aligned and combined into a single image of the field called an orthomosaic. The software then converted the two-dimensional images into three-dimensional point clouds, allowing us to estimate the height of different pixels within each plot. We used UAV imagery taken at two different crop stages to analyze change in pixel height. The first set of the images were captured on the day around flowering stage. At that stage, no lodging had occurred and the crop had reached its maximum height. The second set of the images was captured on the same day as the manual lodging assessments were taken. By that time, lodging had occurred so that the height change in pixel height between two stages of the crop is due to lodging. So we used that to estimate lodging. Here is an example from a single plot. This image shows a two-dimensional representation of the 3D point clouds. The x-axis represents row length and y-axis illustrates the density of the pixels at different heights. The different colors represents height that we slice based on selected percentile heights. The height change between two different stages of the plot is due to lodging. To capture this change, we generated multiple predictors by subsetting height at flight 1 with height of 95th, 85th, 75th, 65th uh, at flight 2 separately. Those predictors were used in various statistical models such as parametric, non-parametric, and ensemble approach. I first learned about the ensemble approach from a presentation by Sunichiro Tomura, and I thought it could be useful here because it combines the information from different models. To select the best performing model, I used standard cross-validation approach and subset the complete data set randomly into training and testing. To evaluate model performance, we use two metrics. One is Pearson correlation and second one, the residual mean square error between observed and predicted plot values. In the corresponding plot, the x-axis shows the models as I mentioned, parametric, non-parametric combination by averaging ensemble, and the y-axis shows the matrix. Each block dot represents the accuracies from one of the 100 iterations, and red dot shows mean across 100 iterations. We are looking for the model with high correlation and low error. When comparing all the models, we observe that in general, non-parametric models outperform parametric ones. However, our study found that the ensemble naive average performed slightly better than the other individual parametric and non-parametric models. This is because each individual model captures different aspects of the data. Some of the features are well predicted by one model and some of them are poorly by another model. By averaging the predictions, we are leveraging the strength of all individual models, resulting in slightly better predictions than those of the individual models. The ensemble model was selected for further analysis. Then I looked at across whole data set to identify plots that were poorly predicted. On y-axis, we can see the residual squares between observed and predicted values. On y-axis, I made those lodging counts into the groups to better understand what's happening. The figure includes results from multiple cross-validation iterations. So single cluster may represent repeated predictions for the same plot. However, some plots showed very large difference between observed and predicted lodging values. These poorly predicted plots appeared as potentially outliers. Therefore, we set a criteria to identify those plots for further visual inspection. Using the criteria, we identified 180 plots as outliers of total 2,675 plots. We had photos of every individual plot, so we compared the plot images with the recorded manual scores. Then we found two main issues from this visual inspection. The first issue related to the plots that were clearly scored incorrect in the field. For example, in one plot, image clearly showed severe lodging, and both uh, the manual scores and UAV predictor scores were 90 percentage, which was correct. However, in another plot, the image showed no lodging, but the recorded manual score was 90 percentage, while the UAV predicted score was 0 percentage. Therefore, this suggested that some field scores may have been incorrectly recorded or assigned. Overall, 82 out of the 180 visually inspected plots appear to be affected by this type of scoring inconsistency. The second issue involved plots with poor germination. In some plots, germination was very poor, so there were few plots and more visible ground. When we looked at plots with high residuals, we found that many of them were poor germination plots. In practice, in practice, in breeding programs, breeders would usually remove those plots from data analysis because poor establishment can compromise lodging assessments. After identifying these two sources of error, scoring inconsistency and poor germination, I wanted to test whether removing those plots would change model performance. The first approach we used, that is versional data including all plots. The second approach removed the plots identified uh, as manual scoring inconsistence. The third approach removed the poorly established plots. The fourth approach removed both manual scoring inconsistency and poorly established plots. Finally, the fifth approach removed both types of problematic plots and then retrained the model. Here, I used residual mean square error to compare the approaches where value closer to 0 indicates low error. What we can see is that removing problematic plots reduced the error, but retrained model after removing them did not uh, improve much. This suggests that the ensemble model was relatively robust even when some scoring inconsistencies and poor establishment plots were present in the dataset. After selecting the model, we looked at factors that may affect prediction accuracy. First, we tested whether the model performed consistently across sites and series, means there are different germplasms I have in my data sets. The results showed no major differences that suggesting prediction accuracy was stable across sites and trial series. Then we compared uh, sampling strategy using two different training set sampling One is stratified sampling, another one is random sampling. Stratified sampling provided only limited improvement compared with the random sampling, suggesting that prediction accuracy was generally stable across even the uh, sampling approaches. So far, I have measured lodging at the plot level to evaluate how well we can predict lodging using UAV imagery. The results showed reasonable prediction accuracy, but observed and predicted values were not identical or not perfect. However, plant breeders are mainly interested in genotype ranking, identifying which genotypes are best and which are the worst. Since manual and UAV phenotyping may produce different rankings, we also need to evaluate how reliable the UAV based method is for breeding decisions. To do this, I conducted multi-trait and multi-site statistical analysis that includes genotypes in the model. I also looked at single trait and multi-site analysis. They were consistently showing similar results. That's the reason here I'm going to show the results for only multi-trait and multi-site analysis. I'm going to present the results for trial series 1 and the results for the other trial series were very similar. One of the things that plant breeders are interested in is heritability, which tells how much of the observed variation is associated with the genetic differences among genotypes. The heritability estimates from the two phenotypic methods were very similar within each site. This suggests that UAV-based predictions captured a comparable proportion of genetic variance to the manual phenotyping approach. However, similar heritability alone does not tell us whether the two methods ranking genotyping in the same way. Therefore, we also looked at genetic correlations between two phenotypic methods, and I'm comparing here within the site. High genetic correlations within the same site indicate that the genotype rankings from two different methods are very similar and very strong. This is what we expected because these two sites were used for model development. However, we also want to know whether the model has broad applicability behind the sites used to develop the model. Therefore, the validation side, we looked at validation side that showed low heritabilities and moderate genetic correlations compared with the model development sites. However, uh, the same general patterns were observed for both visual scores and UAV based phenotypes. Breeders use selection differential to estimate the effect of making selections. Therefore, I selected top 10% of lodging resistant genotypes. Then the selection differential was calculated by subtracting the overall population mean from the mean of top 10% of selected genotypes. Our results showed consistent and comparable selection differential between phenotypic methods within each site. Even at the validation site, selection differentials were comparable. This is providing confidence in the utility of the UAV-based approach for current sorghum breeding programs. From a breeder's point of view, these two phenotypic methods are equivalent. But for plant breeders, it is not only about accuracy, it is about what it costs to run the experiments. For example, the validation site includes 2000 plots. Visual scoring would cost to score those 2000 plots approximately $1,200. If we use UAVs, it is tenfold cheaper. These cost savings could be used to phenotype more locations or we can increase our trials. which could increase selection intensity and contribute to enhanced genetic gain. So far, we have shown that UAVs can replace manually measure lodging, and it allows us to measure more plots at low cost. The next question is whether UAVs can provide additional information to help us to understand this trait better. One opportunity is to take repeated measurements through time. By collecting UAV imagery at multiple time points, we can study how lodging progression over the season and evaluate whether this information can be useful for a breeding program, because in practically lodging changes over time. If I go and measure lodging today and if I go and measure lodging after a week, I will get different answer. Static phenotypes are taken at a single time point. Dynamic phenotypes are taken over a period of time. Here is an example of why this is important. We can measure biomass at the end of the season, but the important thing is for physiologists is to understand how that biomass accumulate over time, that progression. And breeders are also interested in average lodging across sites. As I said previously, in beginning of my presentation, Australian sorghum growing environments are very variable. That's the reason we observe high variability in lodging across sites. Some sites show low lodging, some moderate, and some high lodging based on our visual scoring. If we look at single site, it may provide only limited discrimination among genotypes. In a low lodging site, most of the genotypes look at the end, and if we look at high to moderate to high lodging site, several genotypes look similar at the end. However, when we combine information from multiple environments, we obtain a clear spread among genotypes. This improves genotype discrimination and provides breeders with more useful rankings for making the selections in a breeding program. At that single site, we can measure lodging using the difference between flight 1 and flight 3. That's what I used in my first part of the research and estimated UAV predicted lodging at the plot level. Perhaps if we have information from additional flights taken between flowering and the final time point, we can extract additional information that might help us achieve better discrimination of genotypes. This is a simple example showing why repeated measurements may be useful. At the time point 1, both genotypes show 0% lodging, and at time point 3, both show 100% lodging. So using only the static final phenotype, we can't separate those genotypes. But if I have a middle time point, genotype 1 has already lodged completely while genotype 2 is only partially lodged. This shows that repeated UAV measurements can capture lodging progression and may provide additional information behind static phenotypes. So this part of the research is still ongoing, so I will not go into details about what data I have and what results I have. Instead, I would like to briefly explain the next steps in this part of my PhD. So far, I have shown that UAV imagery can be used to estimate lodging, and it is useful for breeding programs. The next question is whether additional UAV flights can provide extra information behind a single final lodging score. Firstly, I will evaluate whether lodging information derived from additional UAV flight can improve genomic prediction of lodging in historical sorghum trials compared with using endpoint information. Second, I will assess whether correlated traits, including plant height, flowering time, leaf senescence, can improve genomic prediction of lodging. This is important because these traits may help explain difference in lodging risk among the genotypes. The first key message is that UAV based lodging estimates have utility as a phenotypic tool. They provide comparable information to conventional method, which is manual approach, but which is much lower cost. The second key message is that lower cost UAV phenotyping allows us to collect repeated measurements across time. This creates an opportunity to estimate dynamic phenotypes, which may help us capture lodging progression and improve genomic prediction of lodging. I'm thankful to my supervisors for their support, guidance, and valuable inputs throughout my PhD. I'm also thankful to the contributors, reviewers in my progress reviews, and also Sorghum breeding team, because I'm working on existing data, so that data collected by Sorghum breeding team. And I'm also thankful to the ARC Plant Success, IRTG, GRDC, Advanta, and Queensland government, because I'm working from there. Thank you very much for listening my presentation. I'm happy to discuss any questions or comments on the work I have presented today.